the experimental model of fibrosis induced by learning is not from our program, but so it's an opportunity. She's from the Department of Microbiology.
No, from dermatology.
Okay, good afternoon. Thank you for the coordinating the invitation. I'm going to present the project of my PhD. And really didn't have any results. And I'm going to present more than the project the research line of our group. I am supervised by the Professor Carlos Peleschi Taborda, present here. I am a PhD student on the program of dermatology. Fungus have been described more or less 100 years ago, since 1839, dermatomycosis was described, until 1910, Paracoccidioides was described by Adolfo Lutz in Brazil. But it's a little bit that is known about fungi and mycosis really now. One of these fungus are part of the phylum Ascomycota, that combine a group of systemic dimorphic fungi, two Paracoccidioides species, Lucian brasiliensis, Blastomyces, Histoplasma, Coccidioides, Penicillium magnetis, Protrisus schenki, that are dimorphic and include Aspergillus and Candida. Dimorphic fungi reproduce as either by mycelia and yeast-like forms. General mycelia is a prophytic form that grows to 20 to 25 grades centigrade, and just like pathogenic form to 35 grades. This dimorphism is important to the identification of the mycosis to make identification of the diagnosis of the fungus. Generally, the mycelia, as a protective form, grows to the environment temperature and produces spores that are associated to the mycelium. Under the nutritional deprivation, this situation will be reproduced at the laboratory when the culture are deprivated or are poor in nutritional media. Present tinifa with macro or micro conidia that are infecting phase that could be measured until 15 microns depending of the kind of fungus. The yeast-like pathogenic form grows at 35 grades. They are associated to high disponibility of nutrients like infected tissue and the laboratory conditions in rich cultures. That are the parasitic phase that could be measured more than 10 microns by the case of the coccidioides in the tissue. The infected particles, conidia, or portion of the mycelium are produced by the mold that is inhalated and reached to alveoli and from the susceptible host. There are the capacity to transform to the yeast, that is the pathogenic form, and this capacity made this fungus like pathogenic agents able to produce mycosis. The balance between the host and the pathogen made the combination, adequate combination, to generate disease. The host is important in the host to know the immunological status, the lung integrity, and the fungi, the quantity of inoculum, and the virulence of the strain. Paracoccidioides is produced by Paracoccidioides, that is knowledge until now. The species Paracoccidioides brasiliensis and Paracoccidioides lutzi is distributed from north of Mexico until the north of Argentina. The major cases are located in Brazil, Colombia, and Venezuela, are associated specific environment areas, and the host associated in the nature are the armadillos. The mycelium produce conidias, a little bit spores that will be inhalated, and the host could be produced two clinical forms, the acute form or the chronic form, that could be disseminated in mucous membranes, skin, lymph nodes, and adrenal glands, and other organs like the spleen and lung and liver. The treatment of paracoccidioides will be made by itraconazole, amphotericin to the cases more compromised, and sulfamethoxazole trimethoprim. This is the treatment more used in Brazil. And 60% of the patients with PCM could have residual fibrotic lesions. Aluna have a lot of antifungal drugs to treatment the paracoccidioides and the other mycoses. In paracoccidioides are knowledge that have resistance for more of them and have resistance of them to amphotericin B, which is important to knowledge the epidemiology, but mycosis, and this mycosis not is compulsory, not is obligatory information from this health system. Paracoccidioides mycosis occurs the first level and are more than 50% of the deaths caused by systemic mycosis in Brazil. But, however, didn't have any correlation with is associated with AIDS or HB patients. How was talked infection and disease will be a balance between the human virulence factors and the stage of the host. The study of the immune response in the PCM during the initial and chronic stage is so difficult to be the lack of knowledge of natural habitat of the fungus and the time of infection occurred in the patients. It is the reason because the experimental models were developed as alternative to understand to immune response and the problem of the disease. Several models have been described to study the PCM. All of them have different mice strains, fungal stage, the yeast, mycelium, and conidia form, different fungal strains, or inoculation process. From these models, we can analyze similarity. The most similarity is that after a week of infection, they present the highly percent of granulomatous at the lung. That granulomas could be made of kind of parenchymatous, nodular, perivascular, or pseudotumoral granulomas. The treatment, how was talked, will be made by antifungal drugs, but have some immunomodulators that will be tested, like pentoxicillin, that is an antifibrotic drug approved to treat for other kind of patients, that could be used as the models and reduce the granulomas and the fibroses in the model. Another immunomodulator is the use of the P10 peptide of 10 peptide 10 that are part of GP43, that is the major immunoglobulin antigen that are the principal protein for diagnosis of the PCM. Its sequence have 5 amino acids that are the epitope to the T cells. And these T cells offers effective response, TH1, that will be secretes interferon gamma and interleucin 2. The use of P10 for PCM will be used as well in cases of allergy, in animal allergy, that concomitant and together with itraconazole could be decreased the formation of granulomas and dissemination to spleen and liver. As well, the use of P10, the peptide P10, associated with some adjuvants like completed free adjuvant or flagellin or alumin or cationic lipid, could be decrease the load, the burden load of CFU at the lungs. The principal aim of our project is evaluated peptide 10, like a monotherapy or in combination with antifungal drugs in the fibrosis experimental murine model induced with Paracoccidioides conidia. We pretend to analyze the improve of the lung lesions by the observation of numbers of percentage of granulomas formation, the modification of the fibrosis deposition in the tissue, and the cellular acetylkines deposition. All of these in focus before to begin testing with humans or regulators and improve treatment, prevent or diminish or avoid fibrosis lesions that are important sequela in PCM patients. All this model is important to test. The group will work at the acute phase, but it's important to know what is the effect of the peptide to the fibrosis and the chronic form of the disease. Thank you. Thank you very much. Any questions?
So I have a question. The 5 amino acids, the peptide is 10 amino acids long, right? Or
15.
longer? 15. But you've mapped the epitope down to 5?
5.
I mean, it's pretty short.
It's very short.
Have you tried, is there any variation within that epitope if you look at the sequence of a bunch of Paracocci isolates?
Yes. Until now, are acknowledged 2 species of Paracoccidioides, Paracoccidioides brasiliensis and Paracoccidioides lutzi. Have a little bit difference between them at the region of the epitope of the T cells. But until now, our line of research is focused to look if the same peptide will be important to treat the paracoccidioidomycosis caused by Paracoccidioides lutzi.
Okay. How did you choose this one little epitope out of the whole protein? Why not just squirt the protein in? What's the...
No, the peptide in general will be changed in some amino acids. And this part of the amino acid was the most important epitope to T cells. But the other parts, not that important. And the region of 15 amino acids are the protective peptide to the disease protection. Protective peptide.
Yeah.
No sé, Carlos.
Yeah, that's right.
Yeah, that's correct. And we found we did a peptide kinase, and then we discovered the 10 most important peptides used in vaccination. And we also found that there is a part, the 5 amino acids. If you have any mutation, they lost the activity against cannot activate T cells.
Okay, interesting.
But also in Lutzi, we have it's a new species. And Lutzi has a very important mutation and in P10. We don't know if it really will work, and P10 will work as well with brasiliensis and the same time, because there is a mutation in the core. We know we did mutation in the core. Every time that we move any amino acid, we lost the activity. But we don't know if it will work or not with the Lutzi. However, P10 has an adjuvant activity, and maybe it's important to produce interferon gamma. And if then even produce of interferon gamma and Lutzi infection, we can sometime maybe control the disease. We don't know yet.
So one more question. The work you're doing is in mice, right?
Yes.
So the mice. So would the humans? These aren't transgenic mice? HLA transgenic? Are they? Would the humans recognize the same peptide?
Yes, we did. We want to talk.
Fine.
Now the problem in Brazil, and it's very difficult to try in patients. We've done everything. The next step we should try in human and see what happens. We know only in vitro experiment where we know that the P10 is very promiscuous peptide and is recognized more than 80% of HLA, human HLA. And then in vitro experiment works very well. But you should test in human. And you are looking for financial support and to try and see if it will work in human also. And now we have only mice.
Yeah.
Unfortunately, only mice.
But the problem with Paracoccidioides luxi is because it's a new species. So recent to describe it like 5 years, 8 years ago, and didn't have a good classification of the isolated in Brazil to test all the regions of the GP43 protein and the characteristics of the P10 or in all the, because have a like 10 or 15 isolated that is why we are working with them. But it's important to have a mapping of the GP43 from the all isolates. That is something. Thank you.
Thank you guys, everybody for the presence, and so for those who are. We'll try to do the bye-bye at 9 o'clock. Okay, thank you.